You will use an apparatus like this one to perform small-scale dry column vacuum chromatography. You will gently insert a pipette chromatography microcolumn into the hole of the rubber adapter at the top of the apparatus. Don't use too much pressure when inserting the column. Push it just enough so that it's secure and makes a seal. The preparation of the microcolumn is demonstrated in another video. See the experimental procedure for instructions on how to load onto the silica at the top of the column the material that's to be chromatographically separated. The apparatus is connected to a vacuum source via thick-walled rubber hose. When vacuum is applied, the liquid mobile phase that passes through the column will collect in the separatory funnel. You will then break the vacuum, allowing you to drain the liquid from the separatory funnel. This process is now demonstrated. First, close the separatory funnel's stopcock valve. Then attach the pinch clamp to the rubber hose on the T-connector to create a closed system within the apparatus. Then fully open the valve of the vacuum source to create a vacuum. You can then use a pipette to add the mobile phase, also called the eluent, to the top of the loaded column. Let's assume that this is the eluent to be used for the first column fraction. The vacuum will pull the liquid rapidly through the column, and the liquid then collects in the separatory funnel. Once the liquid passes through the column and the column is mostly dry, which should take no more than about 30 seconds, you should remove the pinch clamp from the rubber hose on the T-connector. Then close off the valve at the vacuum source to stop the suction, and then drain the column fraction into the appropriately numbered test tube, in this case number 1, since this is the first column fraction. Don't forget to close the stopcock valve after each time you drain it. Note that you do not have to clean the separatory funnel or any other part of the apparatus after each new fraction is collected. You can proceed directly to adding more eluent to the top of the column and collecting the next fraction in the same manner that you collected the others. What follows is a demonstration of dry column vacuum chromatography used to separate the carotenoid and chlorophyll pigments in a sample of dried parsley. The pigment extraction and preparation of the pipette microcolumn is demonstrated in another video. Gently insert the pipette microcolumn in the rubber adapter, pushing down only hard enough so that the column is secure and makes a seal. Make sure the separatory funnel stopcock is closed, and apply the pinch clamp to the rubber hose of the T-connector. Then open the vacuum valve in your fume hood. The system is now under vacuum, and you can then wash the column with approximately 3 milliliters of heptane, which helps to pack the silica gel of the column more tightly, leading to better chromatographic separation. Use a pipette and rubber bulb to transfer the heptane to the column. The heptane passes through the column fairly quickly. When very little liquid emerges from the column, about 20 to 30 seconds after you added the last portion of heptane, you should remove the pinch clamp from the rubber tube of the T-connector to break the vacuum. Then close off the vacuum valve in your hood, and then drain the heptane from the separatory funnel receiver into a small Erlenmeyer flask. Close the stopcock, cork the flask, and put it aside. You can now get ready to apply the pigment extract to the top of the column. First, attach the pinch clamp to the rubber hose of the T-connector, then open the vacuum valve in your fume hood. The system is now under vacuum and you can transfer the pigment extract to the column using a pipette and rubber bulb. Gently poke the pipette tip through the undissolved solids to the bottom of the tube to draw up the liquid. Don't worry if a small amount of solid is also taken up into the pipette along with the liquid. Apply the liquid directly to the sand located at the top of the column. With vacuum still being applied, use a small amount of the heptane you set aside in an Erlenmeyer flask, no more than 5 to 10 drops, to wash down any of the pigment solution coating the inner glass surface of the column or remaining in the layer of sand above the silica gel. Do this before you begin to add any eluent to the column. At this point you can already see the yellow carotenoid pigments separating away from the green chlorophyll pigments that remain at the top of the column. You can now begin to apply the first eluent to the column. In this case, it's 2 milliliters of a solution that's 9 parts heptane to 1 part ethyl acetate. So that you can more clearly see the descent of the yellow carotenoid pigments down the column, which can sometimes appear as a faint yellow band, a black background has been placed behind the column during the application of the first eluent. Here you can see the yellow carotenoid pigments rapidly moving down the column while the green chlorophyll pigments remain immobile. After adding all of the first eluent, remove the pinch clamp, close off the valve to the vacuum source, and then drain the column fraction into a labeled test tube. Here you can clearly see the yellow pigments that eluted from the column. Set the tube aside safely in a test tube rack. You can see that the green chlorophyll pigments remained at the top of the column after application of the first eluent, which was 9:1 heptane to ethyl acetate. By switching to an eluent with a stronger eleotropic strength, in this case pure ethyl acetate, you'll be able to elute the more polar chlorophyll pigments off the column and into the separatory funnel receiver. Once again, place the pinch clamp on the rubber hose of the T-connector, open the valve to the vacuum source, then apply the eluent to the column using a pipette and rubber bulb. The green pigments can be seen to rapidly move down the column. And they collect in the separatory funnel receiver. Remove the pinch clamp so that the system is no longer under vacuum. Close off the valve of the vacuum source. And then drain the column fraction into a labeled test tube. Here you can clearly see the results of column chromatographic separation of the yellow carotenoid and green chlorophyll pigments extracted from parsley. The chromatographic separation was greatly accelerated by the process of dry column vacuum chromatography.